Morning. Let me first thank the organizers for inviting me here. It's a great pleasure to be here and talk about my work. And I go with June. I have a 4-year-old lab at Hopkins. We do theory and computation, and we love to collaborate with experimentalists. And I am going to talk about today the biophysical modeling of condensate dynamics, bridging theory and experiments. Okay, so as this is my first time here, let me briefly introduce recent research in my lab. Our lab mainly focus on biomolecular phase separation, and in these 3 topics here, we are very interested in how the microscopic features of the biomolecules, as well as their interactions with the environment, impact their phase separation propensity, as well as the condensate physical properties. Now, we also have a branch outside of the condensate field where we are using statistical mechanics and deep learning to understand how mechanics of short DNA segments depend on its sequence. So today's talk, I will mainly talk about our efforts to understand the dynamical aspect of the condensate. So here I'm showing animations of a membrane bound organelle on the left. It's membrane bound, and on the right, it's a biomolecular condensate. I hope you can appreciate the openness of this biomolecular condensate. Now, without this membrane, the molecules inside the condensate are constantly exchanging with molecules in the surrounding dilute phase, whereas in this membrane-bound organelles, molecules remain inside. So condensate is a more flexible and a loose structure in this sense. Now, why do we care about condensate dynamics? We care about the dynamics because it matters to condensate function. The advantage of not having a membrane is that they form and dissolve quickly in response to cellular signals, such as the formation of these signaling clusters and stress granules. And by modulating condensate dynamics, for example, by tuning the availability of substrates and enzymes or the escape of products, cells can then control the biochemical reactions happening inside the condensate, such as the ones that happen inside the nucleolus, this dish I was just talking about, as well as the purinesomes. Okay, so more generally, the material exchange of the condensates with the surrounding dilute phase can also impact the coarsening dynamics of the condensate, which can subsequently impact the number, size, and the locations of the condensates in the cell. And as you can see that these different types of condensate have very different number and sizes, right? Although the thermodynamic equilibrium expectation is that we always have a single merged condensate. Okay, so in this talk, I will really focus on understanding the exchange dynamics of the condensate. And I will first lay out the problem of interest, what is the exchange dynamics I'm talking about. And then my talk will follow by 2 parts. In the first part, we developed a theory for the exchange dynamics of condensate, which was inspired by FRAP experiments on LAF1 protein condensates. And in particular, we found that interface incorporation can be slow and can limit the exchange dynamics. And in the second part, this is a new work where we developed a modified theory for the exchange dynamics, and which was inspired by FRAP experiment on DNA nanostar condensates. And in particular here, we found a pore-mediated pathway which can speed up the exchange. Okay, I'll then end with conclusions and outlook. Okay, so here is the problem of interest. We have a condensate surrounded by dilute phase. Molecules are diffusing inside and outside of the condensate. The only difference is that inside the condensate, we have a much higher concentration. Okay, and the simplest question we can ask here is how long does it take for a droplet to refresh its material by exchanging with the dilute phase? Okay, so experimentally, there are generally two methods to measure the exchange dynamics. One is the fluorescence recovery after photobleaching, the FRAP, and the other is single molecule tracking. And we have developed a theory to interpret both methods from data from both methods. But in this talk, I will mainly talk about the FRAP experiment side. So let's talk about FRAP here. In a FRAP experiment, people usually bleach a small region in the droplet to tell if the droplet is liquid-like. Now, to measure the exchange dynamics, we will have to bleach the entire droplet and record the recovery intensity in the droplet as a function of time. So the droplet is bleached here, and it gradually recovers its intensity through exchange of materials with the dilute phase because the dilute phase molecules are not bleached, right? So we would like to know what controls the time scale of this recovery. That is, in fact, the same time scale as the molecular exchange. Okay, so this is a time scale, the intensity recovered, and we want to know the time scale. So let me start part one of this talk first. Here, this is in collaboration with Ned Wingren, Howard Stone, and Cliff Brangren at Princeton. Okay, so I'm not going to talk about a detailed theory in this part, but simply just highlight our results and physical intuitions. Okay, so what would be your intuition, right? Since molecules are diffusing both inside and outside of the droplets, our first guess was that the recovery time should be controlled by diffusion in the dense phase and dilute phase, right? So indeed, from simple version of theory, I would call it version one, we derived an analytical expression for the recovery time, and which is clearly set by two terms. Okay, the first term here comes from dilute phase diffusion, and the second term comes from dense phase diffusion. Okay, and because these are from diffusive processes, we have characteristic R squared over D dilute and R squared over D dense as the time scales. Now, we also have a prefactor here in the dilute phase term. And this is because we are replacing bleached droplet at a concentration that is C dense by the dilute phase molecules at a concentration of C dilute. Okay, so these are the two rate limiting processes, right? If the dilute phase concentration, C dilute, is low, then the exchange time is limited by the flux coming from the dilute phase, whereas if the internal mixing is slow, then the exchange is limited by the mixing speed with the within the droplet. Okay, so this is a pretty nice theory, I would say. However, our result cannot really explain this FRAP experiment on a LAF1 condensate. Okay, so first from the snapshots, you can see that this LAF1 droplet recovers over the time scale of an hour. Now, when we plug in these experimentally measured parameters into our analytical expression, we find that it only takes 4 seconds for the dilute phase flux to replenish the droplet, and it only takes 60 seconds for internal mixing to happen, right? So clearly 1 hour is not 64 seconds. There must be more going on here. All right, so we thought if it is not the dilute phase and the dense phase diffusion, then maybe it is the interface, right? Because there is only that many things we can think of. Maybe the dynamics at the interface is slow. That is, there is an interface resistance resisting the molecules to cross the interface. And to model this interface resistance, so in our theory version 2, we introduced a phenomenological parameter kappa to control the net flux crossing the interface. And this leads to a third term in the recovery time expression here. So when kappa is small, the flux going through the interface can be rate limiting, leading to a large FRAP recovery time. Note that this interface limited term is proportional to R instead of R squares. And this is because the flux is proportional to the surface area, and the number of molecules to be replaced is proportional to the droplet volume. So R cube over R square makes it linear with R. All right, so coming back to the left one droplet, plug in the numbers, we have obtained a value for kappa here. And to show that this term can indeed dominate the exchange time, we have further performed coarse-grained molecular dynamics simulations with sticker spacer polymers, and which making it essentially an in silico FRAP experiment. So what we found here is first, yes, it is possible to design molecules with their condensate exchange dynamics limited by the interface term. And second, we found that the reason that the interface term can dominate is because incident molecules, they actually bounce at the interface, which slows down their incorporation into the dense phase. Okay, so this is the logic here. Now, I'm of course very curious about what is there in the LAF1 protein that leads to their slow incorporation, but that is more an experimental question. So in the following, instead, I will tell you about an even more surprising FRAP result of DNA nanostar condensate, and also our attempts to understand that. Okay, so now we are moving to part two. In this work, we are in collaboration with Rebecca Shulman and her graduate student, Ali Kangamana at Johns Hopkins University. From the sketch here, so a DNA nanostar is a star-shaped structure made by joining complementary DNA sequences. And this is a nanostar with 3 arms, and these single-stranded end region, they can bind to each other. So they form stickers. They bind to each other to promote phase separation. And micrometer sized droplet can be formed under the microscope for these kind of systems. Right. So Ellie performed FRAP experiments on droplets of DNA nanostars of different sizes. And to our surprise, for droplets of small sizes ranging from 0.8 to 1.5 micrometer, they essentially recover at the same time. So these normalized intensity curves, they are directly extracted from raw images. We do not even do any fit. And you can see that they almost overlap. They just almost overlap with each other. And Ali has also repeated the experiments on different days and verified that this is reproducible. So if you still remember, we have been talking about the recovery time is proportional to R squares if it is diffusion limited, and linearly proportional to R if it is interface limited. But here we are having the recovery time independent of droplet size, which is surprising. So now let me show you that also that for large droplet, the recovery time does increase with R, but there is a flat region, and we saw that maybe this flat region can give us some insights about the dynamics of these DNA nanostars, right? So we wanted to figure out why is this, and motivated by these results, we developed a version 3 of theory to model the exchange dynamics of condensate. So here is our modified picture. Now the theory part was done by a talented graduate student in my lab, Ross Kligman. He proposed a modified physical picture of condensate dynamics. So he reasoned that sticker spacer polymers, they are known to form dynamically restructuring networks that go beyond homopolymer phase separation. And often these are referred to phase separation coupled to percolation. And DNA nanostars are almost perfect sticker spacer polymers, so we should actually expect a percolated network inside the DNA nanostar condensate. So in the modified picture, Now, attachment and detachment of molecules to and from the percolating network intuitively gives to different mobility populations within the condensate, right? The low mobility population, we call it species 1, represents molecules that are bound in the network and they diffuse within the network slowly. And the high mobility population, we call it species 2, they represent freely diffusing molecules that are detached from the network. Note that they can be also inside, not just outside. Okay, and the two states can convert between each other through attachment and detachment. And there is no percolated network outside, so we only have species 2 molecules there. Now, if we plot the concentration of the slow and fast molecules, this is how it looks like as a function of the radial coordinates. So this is inside the condensate, this is outside the dense phase, dilute phase. I want to emphasize that like this is a single component condensate. We only have one type of molecules. We've separated the molecules based on their network connectivity and mobility into two populations. But they have the same identical chemical identity. So Ross developed a continuum model to describe, called the reaction diffusion model, to describe the exchange dynamics of the condensates for this modified picture with two mobility species. Now these two equations looks long, but what they really mean is that first, C1 and C2 are just the bleached concentration of species 1 and 2. The first terms here on the right, they are conventional diffusion in a concentration gradient, right? Because without the rest of the terms, these are just the normal diffusion equations. Now the second terms here, they represent excess chemical potentials that drive molecular phase separation, right? Because they are not phase separating, they are not diffusing in a flat space, but actually in a phase separated environment. So this enforces them to have a non-uniform concentration. And finally, we have the third and fourth term here. They account for mobility switching between binding and unbinding between the network. Okay, so if we know the concentration, like these are the equations. If we know the concentration of the equilibrium concentration profiles and the dense phase diffusion as well as the conversion rate, then these two equations together with proper initial and boundary conditions can be solved either analytically or numerically and giving rise to the normalized droplet intensity curve, and which allows us then to extract the recovery time. Okay, so this is overall theoretical framework. And let's first talk about the C2 dense equal to 0 limit, which is to say that there is no free polymers inside the condensate. Everything inside the condensate is class-linked. And in this case, species 1 molecules, these class-linked molecules, they are diffusing inside the condensate, and the species 2 molecules, they are outside, and they can only convert with each other at the droplet interface region. And so we can work out the math, and turns out that the recovery time has 3 contributions: diffusion in the dilute phase, diffusion in the dense phase, and diffusion at the interface. And this is exactly the results we had before in theory version 2. Now, except here, we have a clear physical meaning for the phenomenological parameter kappa we have introduced before. Kappa depends on the attachment rate of a molecule to the network, depends on the dilute phase concentration as well as the interface width. Okay, so moving to the non-zero C2 dense case. Now we have free polymers inside the condensate. They are diffusing. Okay, so let me say that. They are diffusing inside. So we found one more pathway for the condensate to recover, right? The unbleached molecules, they from the dilute phase can sneak through the pores of the network and diffuse into the dense phase as species 2, and then they attach and detach from the network throughout the bulk of the condensate. So they penetrate the interface and directly attach inside. And we refer this to a pore-mediated recovery pathway. And if conversion is very fast, so here in this, like when in the presence of this free population, we have the pore-mediated pathway. In this case, we have two limits. First, if conversion is very fast, which means that the molecules in the dense phase will constantly switch between the network attached and detached states. When the molecule is attached, it diffuses slowly with D1 dense, and when the molecule is detached, it diffuses fast with D2 dense. So over these two states, the molecule in the dense phase undergoes diffusive motion with an effective diffusion coefficient D effect. Okay, so recovery time therefore has two contributions. One is still from the dilute phase diffusion as before. The other one is from the dense phase diffusion. Now, everything looks similar except that the D1 dense previously we have is now replaced by the D effect. And because the D effect is a faster diffusion compared to D1 dense, so this pore-mediated pathway speed up the dense phase mixing. Okay, so just to say that this is another hand-waving argument, but really it comes from a rigorous derivation. And in the upper limit here now, still with this pore-mediated pathway, but now we are talking about the conversion is very slow in this limit. When the conversion is very slow, the recovery time is now controlled by the conversion between the species 1 and 2 molecules. Now, since conversion can happen both at the interface and at the bulk of the dense phase, and these two processes, in fact, happen in parallel. So the conversion limited recovery time then contains two contributions. This part here comes from the interface conversion as before, and this part here comes from dense phase conversion directly inside. So this is from interface, this is from the dense phase, where you have the free polymer and attached detached directly inside. So again, these are expressions from rigorous derivation, and taking together these results, here is the recovery time we have in the presence of this pore-mediated pathway, and compared to our serial version 2, okay, the pore-mediated pathway speed up first the dense phase diffusion, right? We just mentioned that the slower diffusion coefficient is now replaced by a faster effective diffusion coefficient. Also, that let's note that there is a bulk condensate conversion, like there is a conversion inside the bulk of the condensate, which can also speed up the recovery. And in that situation, I want you to pay attention here that if first this term is dominating the whole expression, and also the interface conversion is slower than this bulk condensate conversion, then this R cancels with this R here, making the conversion limited timescale independent of droplet size. And this is interesting. We have a constant now that is independent of radius, right? You'd incline to think that the larger the droplet, the longer it takes to recover, but not in this regime. Okay, so coming back to the FRAP experiments on nanostar droplets, we interpret that this flat regime is bulk conversion limited, which means that this term is smaller than that. And we further used the experimentally measure the parameters to find that this term is also very small. So to the end, it leads to the recovery time as a constant plus a term proportional to R squared. Okay, so let's talk about the physical origin of this constant. Now it turns out to be that it turns out that it is the detachment timescale of the or the lifetime of a molecule in the cross-linked network. So let's think about like this constant actually comes from the case when when it dominates. What it means? It means the when this one dominates, it means that the dense phase diffusion is faster, right? So in this limit, what is happening is that what is really matters here is that the bleached molecules, they have to detach from the network, and then they then diffuse quickly out of the condensate, and the vacancy can then be replaced by unbreached molecules. So that is why you have a constant, because the detachment from the bulk is proportional to volume, and the number of molecules that has to be replaced also is proportional to volume. So volume cancels volume, giving you a constant. So fitting the experimental data to the theory curve, we obtained the unbinding time of a nanostar from the percolated network. We found the unbinding time about 150 seconds, and we can further predict the on rate and effective diffusion in the dense phase for the nanostar system. And these are parameters that can be tested experimentally. All right, so before I move to conclusion, this slide is for experimentalists. How to tell what is the rate limiting process for the exchange dynamics for your condensate you are interested. So first, if you measure the recovery time for droplet of different sizes, we expect different scaling behaviors for different rate limiting processes. Diffusion limited is proportional to R squared. Interface limited is linearly proportional to R, whereas bulk conversion limited is a constant. Second, there is also features in the flap recovery patterns. Different rate limiting processes have different flap recovery patterns. If the internal mixing is slow, then the unbleached molecules will slowly propagate toward the center of the droplet. Whereas if it is not limited by the internal mixing, then the rest of the process, they will lead to intensity inside the droplet will be almost remain uniform during the recovery. So they recover while being uniform inside the condensate. All right, so we are here to conclude. We have derived 3 versions of theory to understand the material exchange between the condensate and its surrounding dilute phase. We found that interface incorporation can slow down the condensate exchange dynamics, but a pore-mediated pathway comes to rescue, which can speed up the exchange. And let me also just briefly mention the biological relevance. So biological condensates are porous. 60 to 80% of its content is water. And biological condensates inside the cell have many components. We expect small components can easily travel through the pores and interact directly with the bulk dense phase. And this is likely to have an impact on condensate function. Okay. With that, I would like to thank my old and new collaborators on these 2 projects, and thank you also for listening, and I'm happy to take questions.
Thank you once again, Zhang, for a wonderful talk. And so we have time for some questions. I'm going to ask them. So the first question is from Guangxi, and they're saying, I'm wondering what controls the conversion rate between the 2 species?
Oh, okay, yeah. So what controls, it's really just like what controls, say, binding of 2 molecules, right? And it can be very different for different types of molecules. For DNA nanostars, I guess it's the searching time, which uh, it's going to be, I guess, the molecule size usually matters. The viscosity of the solvent also matters. Um, and also like specific bond angles that you have to match when you form the complex also matters. Yeah, so these are general guidance, but different molecules will have different requirements. Yeah.
Thanks, June. So then the next question is from Miranda Lynch, and she's asking, the pore-mediated pathway model seems very powerful. The characteristics of the proposed pore network seems to be incorporated entirely via the D effective term. Does the dynamics of the pore network need to be accounted for? is there any benefit from adding terms addressing specific features of the pore network?
Oh, whole network. So I would say like if the conversion is very fast, then yes, essentially the effect of the pore mediated network can be incorporated into uh, the effective diffusion. But if uh, the conversion is slow, then it's a different story. And what when do we mean like fast or slow, right? It really depends on like how essentially it depends on how long can you travel before you attach. And small molecules, their diffusion coefficient is very fast. They can still be diffusing very fast when it's kind of sneaking through the pore. So they could, in fact, like really just before attaching, they can penetrate the whole condensate before even attaching. So of course, this kind of situation is very different from a diffusion problem. Um, yeah, and they should be considered very specifically.
Thank you. Then there's a question from Taimei Lu, and their question is, I was wondering, does this recovery time equation also apply to glass-like or high viscosity condensates? In the meantime, what does pore mediated mean? Does it refer to nanopores or how were the pores created?
Well, so glass state, usually it really depends. I saw the glassy like state may not only just show slow diffusion, but also have heterogeneous kind of uh, special distribution of the dynamics, right? It's like you may be very slow in this region, but faster in that region. So that is not accounted in our theory right now because it's a homogeneous assumption that assume everything is homogeneous inside. We can have two populations, but each population only have one diffusion, not have any spatial dependence. And how the pores are created, or these are not nanopores, these are just like polymers. If um, you like cross link them, they naturally form pores, right? It's like this. I don't know if there are people who's in chromatin side, DNA, they are kind of the same chromatins form dense region and kind of low concentration or low density region. And there are always kind of intermediate uh, gaps in between these that molecules can travel through. So it's the same analogy. I guess the idea I want to highlight is really that condensate is not like a solid structure. In fact, the water is really like 70, on average, 70% is water, which means it's very porous. It's like a sponge. Yeah, so you can travel through many holes. Molecules are not just like have to kind of really attach on the surface, but going all over the place.
Okay, we'll take one more question before we go to the informal Q&A phase. That's from Chaitanya, and they're asking, could you please elaborate on whether the difference and increasing complexity of the models are likely to be functionally different in a cell?
Different in a cell. Yeah,
So
so that's,
yeah,
yeah.
so because your models are increasingly more complex,
Yeah,
right?
yeah,
So yeah.
yeah. Yeah, that's what I was hoping to expand a little bit more, but I know I run out of time. It's about the biological relevance, right? When the pore-mediated pathway is really relevant to biology. Because, in simulation or in other situations, we can create any kind of systems that we want to have the specific feature. But is that related to biology? My sense is that like especially say for chemically reactive condensates, they have to really like have substrate entering and the products leaving, right? And then the size of the substrate and products are very important because when you have porous medium, the pore size and your molecule size, the comparison of the two matters a lot if these molecules can enter, right? If you have big molecules, they cannot enter. If you have small molecules, they can enter very easily. So I feel like having a small substrate could probably benefit to then speed up the chemical reactions. But that said that, well, whether your substrate is small or big is not up to, it's not up to you. Yeah. Yeah.
Thank you, June. We will stop the recording and thank both June and Lishi